In today's practical, we are going to isolate DNA from the given sample. Here we are using banana fruit sample to isolate the DNA. For that, we have certain requirements, like we need banana, one Ziploc bag, we need this salt, liquid soap. We can use any liquid soap. Here I am using this Dettol hand wash. You can use any dishwash liquid or any liquid soap. We need any spoon for measurement, water. We need ethyl alcohol. Here I am using ethyl alcohol. We can use any alcohol, and as it is available in the laboratory, I am using here this. If you are doing the practical at home, you can use alcohol based hand sanitizer also. Also we need container, any empty container, and coffee filter. Forceps. And this alcohol, always we need chilled alcohol, that is ice chilled alcohol we are going to use for the isolation of DNA from this banana sample. So these are our requirements, and now let's start with the procedure. So now first of all, we have to peel this banana. Here I am taking one whole banana. And you have to take this banana into this Ziploc bag. One banana, for one banana, I am using here half beaker of water. So I am taking half beaker of water. Put the water in the Ziploc bag. Remove the excess air. And then seal it properly. You have to seal it properly. And now we have to mash the banana and make the banana mash. Okay, so in this way, by physical means, you can mash the banana and make the banana mash. Physically, easily we can mash the banana. So why we are making this banana mash? So with that, the cells are going to separate. The banana cells are going to separate. Yes. So with the gentle hand, you have to mash the banana. In this way, you can see we are preparing our banana mash. Okay. So here in this way, the cells which are present in the banana are going to separate only by physical means. So we are almost done. Mash it properly. So in this way, our banana mash is ready. Yes. Now keep this aside. This is our banana mash. Keep it aside. Now what we have to do, I am taking here one empty container. In this container, we have to take 1 teaspoon of liquid soap. See here, the measurement should be proper. Measurements should be proper. 1 teaspoon of liquid soap. We need half teaspoon of salt. And we need 2 teaspoon of water. As I am going to make this mix, yes, this practical you all 25 students are going to do, so I will take just double amount. Yes, 1 teaspoon of liquid soap, half teaspoon of salt and 2 teaspoon of water. Again I am taking same, so that everyone will get this filtrate. 1 teaspoon of again, and half teaspoon of more salt and 2 teaspoon of water. So here I took 2 teaspoon of soap, 1 teaspoon of salt and 4 teaspoon of water. Now mix it well, mix it gently, so that the salt and soap can be easily dissolved in this. Mix it well. Now add banana sample, this banana mash. I am going to add 4 teaspoon of this banana mash. 1, 2, 3 and 4. 4 teaspoon of banana mash. You can take little more. Yes. Now mix it properly. Stir it gently. You have to stir it around 15 to 20 minutes. Yes, 15 to 20 minutes you have to mix it. Continuous stirring, continuous stirring. So now we are mixing 15 to 20 minutes. Here I had taken what? Liquid soap, yes, and what? Salt, water and banana mash. Yes, this liquid soap which we are using here, that is going to degrade the membrane of phospholipid and proteins. They are going to degrade the membranes of phospholipid and proteins, so that it will help to release the DNA from the banana sample. Yes. Also we are using here salt. Salt is what? Sodium chloride. Yes, Na plus and Cl negative. So Na plus, what it will do? It will shed the negative charge phosphate group, yes, of DNA, and helping them to separate the DNA in the precipitate form. Okay. Along with that, at last we will add here ethyl alcohol. Any alcohol we can use. Here we are going to use ethyl alcohol. That too ice chilled alcohol. So why we ice chilled alcohol? Why we are going to use ice chilled alcohol? For the separation of DNA from this sample. So this chilling treatment or the cooling effect, that slows the breakdown of DNA. So that DNA will break slowly. Okay. And the alcohol, at last when we will filter this after 20 minutes, so the alcohol will precipitate always in the upper layer. Okay. So DNA, the white layer of precipitate, we will get from this sample. Okay, so DNA will be precipitated in the white color. Okay, white precipitate will be found. So firstly stir it gently 15 to 20 minutes. So now in this way we stirred it well for almost 15 to 20 minutes, and now 20 minutes are done. Now we are going to filter. For that, here I am taking one empty beaker, this coffee filter, and you have to pour this mixture in this filter. In this way, you have to mix here in the filter. Slowly you can see your filtrate is collecting where in the beaker. So almost it is done. Continue. Okay, so in this way you can see this is our what? Filtrate. Now we are going to take this ice chilled ethyl alcohol which I had taken here in the beaker. So this is ice chilled alcohol which we are going to take in the beaker. This is clean test tube. Yes, so take your test tube and almost half of the test tube add this alcohol. So almost you can see half of the test tube alcohol, and that is ice chilled alcohol, and this chilling treatment will help to separate the DNA. And then this is our filtrate. We are going to add this filtrate of the soap, salt, water and banana mix what we had stirred for 20 minutes, and then this filtrate we are going to add in this test tube almost half of the equal amount. This half we had taken this alcohol and half we are going to take this filtrate. Yes, so drop by drop, by touching the wall of the test tube, drop by drop, this way you have to add in the test tube. Slowly, drop by drop, we are adding this filtrate in the test tube. You can see here the alcohol comes at the upper surface, and DNA is going to separate in the form of white precipitate in the test tube. So firstly we had taken half of the test tube ice chilled alcohol, and then we are adding this filtrate, this equal amount drop by drop by touching the wall of the test tube. Almost done. Yes, now we have to keep this test tube undisturbed for 5 minutes. You can see DNA isolation already started, but when we keep it for 5 minutes to settle, then after 5 minutes you can observe the DNA will be completely isolated in the form of white precipitate. Yes, so I am keeping it undisturbed for 5 minutes, and after 5 minutes again we will see our observation. Now almost 5 minutes are done, and you can see DNA is isolated in the sample. Yes, you can see this is what? DNA. So in this way you will get DNA in the form of white precipitate, and here you can see in the test tube your DNA is separated in the given sample. Yes, here we have taken banana sample, and the same way you can use other fruits also, like papaya, yes, mangoes, any other fruit, to extract the DNA from the sample. So in this way we had isolated the DNA from the sample, and here it is isolated in the form of white precipitate. Yes, okay. Now you perform your experiment. Take the sample or filtrate from here. Take the alcohol, an equal amount of alcohol, and put the filtrate drop by drop in your test tube, and you will get your result. Show me, beta. You can see your DNA is isolated. Yes. Nilesh, show your test tube. Yes, very good. You can see here DNA is isolated in the form of white precipitate. Others also doing? Very good. Everyone have to do? Okay, do your experiment and show your result. Yes, show your results. Yes, very good. You can see DNA is isolated in the form of white precipitate. Very good, Kamlesh, Rahul, Bavarlal. Very good, beta. Very nice. All are doing so well. Wonderful. Good.